Nextflow for bioinformatics. Nextflow is a bioinformatics workflow management system designed to make it easier to create, share, and run data analysis workflows. It is particularly well-suited for handling the complexities of bioinformatics tasks, where analyses often involve multiple tools and dependencies. Some key aspects of Nextflow in the context of bioinformatics. 1. Declarative and portable. Nextflow allows you to express your analysis pipeline in a declarative manner, specifying the steps, inputs, and outputs. This makes workflows portable and easily shareable across different computing environments. 2. Dependency Management. Nextflow automatically manages dependencies, ensuring that the required software tools and libraries are available and properly configured for each step in the workflow. This helps to avoid common issues related to software. 3. Parallel execution. Nextflow supports parallel and distributed execution, allowing you to run your workflow on a single machine, a cluster, or in the cloud. This can significantly reduce the time, especially for large-scale genomic datasets. 4. Data handling. Nextflow simplifies the handling of input and output data. It supports a variety of data storage systems, including local file systems, networked file systems, and cloud storage solutions. Reproducibility. By capturing all the details of your analysis pipeline in a Nextflow script, you enhance the reproducibility of your results. The same workflow can be executed with the same inputs, producing consistent and comparable results. Nextflow has a growing community of bioinformaticians and computational biologists contribute to the development of workflows and share them through a centralized repository. This can save researchers time by leveraging existing workflows.